Welcome to the third part of the PamGene data analysis training. In the previous videos, I talked about experiment design, image analysis, and the first part of the data analysis, the basic processing. In this video, I will talk about the downstream analysis, which includes phosphosite and kinase analysis. Let's start with phosphosite analysis. Here is an overview of the downstream parts of the two types of workflows. In the C1T1C2T2 design, we are interested in comparing test versus control in each supergroup, and also sometimes we compare the same conditions between the two supergroups. In this workflow, we use paired or unpaired t-test. In the C1T2T3 workflow, we are interested in comparing multiple conditions versus one control condition. For example, everything versus control, T2 and T3 versus T1, and T3 versus T2. For this, we use the multiple treatments versus control app, MTVC. More on this in a bit. You can also observe that phosphosite analysis is done on log or VSN data and not on batch-corrected data. We usually use combat correction only for upstream kinase analysis. Why is combat not done for phosphosite analysis? If there are batch effects, then we must take them into consideration. This can be done by batch correction or pairing. However, for phosphosite analysis, the best way to counteract batch effects is to do paired tests. Pairing counteracts batch effects, since we run statistical tests within a batch. If there is no batch effect, pairing is not needed. Let's go to Tersin and check how the multiple treatment versus control works. MTVC first does an ANOVA followed by a Dinetz test. Based on the results of the ANOVA, each individual test condition is tested against the control by Dinetz test. This test is a more powerful version of performing multiple separate t-tests. Let's run it. We can see in the wizard that some factors are pre-filled, but some are not. So let's go through each step. First, we select the quantitation type, which is the log transformed. Next is to select which factor defines the peptides. This is always ID, and this is pre-selected in all the apps. The single data value per cell defines the observations. This is defined by barcode and row in most of the cases. We can leave them in. Next, we have to choose what is the control condition. It is the test condition equals control, so I write control here and apply the filter. You can see now why it is important to keep naming conventions. We don't have to think about how exactly our control is named and which factor contains it. It is always test condition equals control with capital C. If you forget though or want to use something else, you can always choose a different factor here and search for the exact values. To search for the values, on some screens we have to zoom out to see the names. Then, we have to click the Apply Filter button and go to the next. Here the question is what is the test condition, and it is defined by a factor called test condition in most cases, so it is pre-selected. The next question is the pairing factor. Since this is a CT1T2T3 design, in most of the cases we have biological pairing, that is, the same replicate of a cell line is used for control and the test conditions. Usually the design is chosen such that one biological replicate is on the same chip. So as pair, I choose barcode here. You can see that colors have appeared based on barcode. However, pairing is optional. The last wizard question is to choose which comparisons we would like to see in the results export file. We have 3 MTVC apps, and in each, all conditions are compared against a different control condition. However, we are not interested in all of them. For example, it is enough to compare T1 to control, and we don't need to compare control to T1. The test is nevertheless run to make comparisons more powerful. But at the end, we have a filter for comparisons to export. This is controlled by this wizard step. In this app, we compare everything to control, so we can leave all the pre-selected conditions in the filter. Let's run the app. The app is run, let's check the visualizations. First, I will show the volcano plot. With this, we can quickly assess how many significant peptides we have in each comparison. You can see the test condition in the columns. However, here the test condition actually means a comparison with the control condition, so T1 is actually T1 versus control. Let's look at the view called ratio map for significant peptides. Here, we have the log fold change plotted for each comparison, for each peptide. We can see the name of the peptides here. To have a bit more insight into which proteins could have been phosphorylated in the sample, we can enrich this data by protein names. First, we need to reset the step. Then, from the image analysis output, we can bring the UniProt name to the rows. Because of some matching problem, some empty data appeared which we can easily filter out by setting UniProt name not equals empty. You can see that for some IDs, multiple IDs match to the same protein, while for some others, the same ID matches to multiple proteins. In this case, we get the same log fold change values for all of them. This view is useful when you want to check what exact proteins in your data could have been phosphorylated. However, we do not measure the phosphorylation of native proteins in your sample, but the phosphorylation of only the peptides on the chip. So treat these results as a possibility of protein phosphorylation rather than evidence. Next, on the other template, I will show how to run the t-test app. This is a C1E1 C2T2 template, and we can check on the heat map how the conditions look like. There is a test and control condition for both supergroup 1 and 2. Let's run this t-test app. It compares test versus control separately in both supergroups. This is one of the things in which the t-test is designed differently than the MTVC. It can work for multiple supergroups at once, but it can only compare one test condition to another. While the MTVC works for only one supergroup, but can compare multiple test conditions to one control. I will go through the wizard steps. I will add test condition as test condition and supergroup as supergroup. For pairing factor, the barcode is already selected. I will leave this in, since test and control are usually from the same replicate, thus there is biological pairing between the samples. You can also see that the wizard doesn't ask for selecting the control condition, as opposed to as in the MTVC app. This is because in t-test, the control is automatically selected. It is the condition starting with an earlier letter in the alphabet. One more reason to keep to the naming conventions. While it is running, let's run the other app. This compares supergroup 2 versus supergroup 1 in both control and test conditions. Here, we don't have biological pairing, since supergroups represent different cell lines. However, when there is a batch effect, it is still good to do a paired t-test. I checked if there is a batch effect in PCA. Here is a screenshot. The different colors are for different chips, and they are not clustered together, so this does not indicate batch effect. We can confirm this in heat map too. I will open the heat map app this time with this icon, not by double-clicking. You can see that I duplicated the heat map step and did a slightly different visualization with barcodes on the top. This also does not indicate any batch effects. There is no barcode which would have generally higher or lower values, for example. Since there is no batch effect, we can do an unpaired t-test. With this app, we have to make 2 modifications for an unpaired test. I will quickly go through the wizard and set the supergroup as test condition and the test condition as supergroup. Importantly, I will delete the barcode from the pairing factor, leaving it empty, because we are doing an unpaired t-test. However, to run unpaired t-test, we have to make one more modification. Oops. The app has returned an error. Let's open it up to see where the error comes from. It comes from the t-test step, since it is red. Let's go inside, reset, and run it again. Now we can see the error message. Error. Labels are required for a paired test. This is a very useful error message, since it tells you that the app still wants to do a paired test, even though we did not fill the pairing question in the wizard. This is because there is one more setting we have to set correctly in this step. Let's go to the Settings tab. Here is a parameter for pairing, which we have to set to false. Now, let's run the step. The step has run, but not the visualizations, so I will run all of them and go back to the workflow. Let's check the visualizations from t-test. They are very similar to that of the multiple treatment versus control. There is a volcano plot, and there is one called significant treatment effects. This is actually slightly different than in the MTVC app, since instead of the fold change, it has the scaled value in the y-axis. Can you recall where we have seen this factor called scaled values before? If we go inside the app, we can see that before the significant treatment effects, there is a step. This again has the z-score operator. I have shown you how the Phosphosite Analysis apps work, the multiple treatment versus control, and the t-test. Let's summarize what you can report from them. First, the volcano plots are good as an overview to check the number of significant phosphosites between your conditions, as well as their direction. Are they downregulated or upregulated, or both? Second, you have log-fold change or z-score scaled input value heat maps. In these, you can see the exact peptide as well as the proteins they map to. If you would like to do further analysis, MTVC and t-test results are also exported to the project library in table format that you can download as comma-separated files, which you can open in Excel. Let's check them. The export files always start with export, then a dash, then the name of the workflow, then a dash, then the name of the app, and then the name of the export step, which is export in this case. Let's check the MTVC files. They are exported in a long format. The variable column contains the log fold change and the p-value, while the value column contains their actual values. More on long and wide format you can find in the first video, Getting Started with Tersin. Let's go back to the project library and check the t-test files. They are exported as a wide format, with delta and the p-value in different columns. Delta is the log fold change. If you would like to export the protein names as well, you have to make a change in the apps. Let me show you. You can open up the app and go to the Select Export step, which just does a selection for exporting. Reset it, and you can change how you want to export. I quickly changed the crosstab views to show you a different way you can export. There are multiple ways, and you can set what works the best for you. If you would like to have the fold change and the p-value in different columns from the ANOVA and DNet step, you can add the log fold change and the p-value treatment to the rows. Next, you can add the protein name or uniprot to the rows, to the left, or to the right from the peptide ID. If you add it to the left, the peptide IDs will be duplicated. If you add it to the right, the uniprots will be duplicated. Here, you can see, for example, that these three peptide IDs match to three AKT proteins, AKT1, 2, and 3, and the values are duplicated. Importantly, because of how the matching is done, you have to filter out the empty data that appears. Here, also the control was filtered out because it has anyway not a number and yen values. Before we go the kinase analysis, I would like to explain the concept of pairing in PamGene data in a bit more depth. I have walked you through the types of pairing and the decisions we can make in phosphosite analysis, but let's summarize it. We can consider two kinds of pairing, biological and technical. The biological is the usual type of pairing when we use the same replicate of the same cell line to make test and control conditions. Usually, the design is chosen as such that the biological replicates and the chips, that is the technical pairing, are the same. However, even when there is no biological pairing, for example, for comparing a cell line to another one, sometimes it is good to do paired analysis for PamGene data. In these cases, we talk about technical pairing. Samples from different cell lines that are on the same chip are technically paired. We generally prefer paired analysis for phosphosite data because it can be more powerful and sensitive. On the right side, you can see a depiction of a case when there is very strong batch effect for barcode. In the PCA, we would see that within each barcode, condition 1 and condition 2 are clearly different, but if we would do an unpaired analysis here, we would probably get less significant results than for paired analysis due to the high variability within a condition. This table summarizes the decisions to make for a simple, one-run design. First, we need to consider whether we have biological pairing between the conditions in the comparison. If yes, we do paired MTVC or t-test, depending on the design. If no, then we still do paired test if we see strong batch effect on PCA. This second table summarizes the decisions for phosphosite analysis for similar designs having more runs. For example, when we have primary cell or patient samples, it is good to have 6 replicates, and then these decisions apply. You can stop the video to read it. If you would like to do experiments with different designs than these, please talk to us, and we can help designing your experiments in the best way possible.